Proteins need to be targeted to specific places within cells, to meet their protein partners, to assemble into complexes or form domains, and to carry out their proper functions. But how does a protein know where to go? Our lab studies novel mechanisms of protein targeting and their importance for function and cell physiology. First of all, a protein may have an intrinsic protein targeting sequence that confers localization, or it may not. A protein may have protein-protein interaction domains, but these alone may not be sufficient. There are thousands of different proteins within cells, so how does one know where to go? Messenger RNAs that encode specific proteins are exported from the nucleus for translation in the cytoplasm. Our work shows that messenger RNAs are targeted to discrete sites within cells for localized translation. RNAs have distinct sequence motifs or structures that allow them to target to specific sites, mitochondria, nuclear or cortical ER, peroxisomes, or polarized domains. Specific RNA binding proteins facilitate the process by binding the individual RNAs and assembling them into large particles that are delivered either along cytoskeletal tracks or by diffusion capture. By transferring individual RNAs, the cellular machinery for protein synthesis, the ribosome, can now make thousands of protein copies from a single RNA. Thus, cells maximize production efficiency by transferring the RNA blueprints to discrete places within the cell and making proteins locally. In our lab, we identify RNA binding and targeting proteins by pulling down individual mRNAs, identifying the bound proteins by mass spectrometry, and then performing mutational analysis. Mutations in RNA binding proteins can lead to RNA mislocalization, protein mislocalization, and defects in essential cellular processes. Thus, mRNA trafficking and the localized translation of protein is an essential and conserved process in all eukaryotic cells and allows for fast cellular responses to extracellular signals.